What if a drug already approved for multiple sclerosis could also help restore the brain's own ability to suppress neuropathic pain? That is the central idea of this paper. The study suggests that Ozanimod, a functional antagonist of sphingosine 1-phosphate receptor 1, or S1PR1, not only reduced pain-like behavior in rodent models of neuropathic pain, but may also act at the level of the brainstem, specifically the rostral ventral medulla, to re-engage descending inhibitory control. Clinically, that matters because neuropathic pain is often understood as a problem of peripheral nerve damage or spinal sensitization. But this paper reminds us that failed top-down pain control may also be part of the disease state. Hi, I'm Dr. Akihiro Ando from Tokyo, and I specialize in orofacial pain and oral medicine. Today, I'd like to review a very interesting 2025 paper published in Pain. The authors examined whether S1PR1 signaling in the rostral ventral medulla, or RVM, contributes to neuropathic pain, and whether Ozanimod can reverse that process. This is an important question because previous work had already implicated the S1PR1 pathway in the spinal cord and peripheral nervous system, but the supraspinal role had remained unclear. The clinical relevance is easy to see. Neuropathic pain remains difficult to treat, and many currently available options provide incomplete relief or unacceptable adverse effects. So when a pathway is already pharmacologically targetable and the drug is already FDA approved for another indication, that immediately raises the possibility of repurposing. Of course, that does not mean clinical efficacy is established, but it makes the translational question much more compelling. The authors used several male rodent models of neuropathic pain. One was chronic constriction injury of the sciatic nerve, which is a traumatic nerve injury model. The others were chemotherapy-induced neuropathy models using oxaliplatin and paclitaxel. These models are useful because they represent different causes of neuropathic pain, and that helps test whether the effect is narrow or more general. First, they ask whether systemic Ozanimod reduces pain-like hypersensitivity. In mice with chronic constriction injury, a single intraperitoneal injection of Ozanimod reversed mechanical allodynia in a dose-dependent and time-dependent way. Then in rats treated with oxaliplatin, systemic Ozanimod both prevented the development of mechanical and cold allodynia and reversed these hypersensitivities after they were established. Similar preventive effects were also seen in the paclitaxel model. So at the behavioral level, the drug showed fairly broad anti-nociceptive effects across traumatic and chemotherapy-related neuropathic pain models. That alone is interesting, but the more novel part of the paper is the brainstem story. The RVM is a key relay in descending pain modulation. It can facilitate pain, but it can also inhibit pain depending on the balance of activity within its circuits and its downstream effects on the spinal cord. The authors directly injected Ozanimod bilaterally into the RVM and found that this local administration reversed mechanical allodynia in rats with chronic constriction injury. They saw a similar reversal in the oxaliplatin model. This strongly suggests that S1PR1 signaling in the RVM is not just a bystander, it appears to contribute functionally to the maintenance of neuropathic pain. The next question is how? To explore a mechanism, the authors performed RNA sequencing of RVM tissue. After nerve injury, they found substantial gene expression changes with a particularly notable downregulation of immune-related pathways, especially type 1 interferon signaling. That is a striking observation because type 1 interferons are often discussed in relation to antiviral biology and inflammation, but they may also influence pain processing in context-dependent ways. In this study, chronic constriction injury was associated with downregulation of a cluster of interferon-related genes in the RVM, including genes such as IRF7 and PTPN6. When Ozanimod was given into the RVM, many of these injury-related changes were reversed. The authors interpret this as evidence that S1PR1 activation in the neuropathic state may suppress an interferon-centered immune axis in the RVM, and that blocking S1PR1 helps normalize this network. The proposed model is not that Ozanimod simply numbs the system, but that it restores a dysregulated supraspinal control mechanism. Now that mechanistic interpretation is interesting, but we should be careful. RNA-seq shows association and pathway-level changes. It supports a biologically plausible model, but it does not by itself prove a direct causal chain from S1PR1 to interferon signaling to behavioral recovery. The paper does provide supportive validation with quantitative PCR for selected genes, which strengthens confidence in the transcriptomic signal. Still, this part of the story should be seen as a strong mechanistic hypothesis rather than a fully closed loop. The pharmacology of the descending pathway is another important strength of the paper. The authors asked whether the analgesic effect of intra-RVM Ozanimod depends on descending inhibitory projections to the spinal cord. They used intrathecal methosergide, a serotonin receptor antagonist, and intrathecal yohimbine, an alpha-2 adrenergic antagonist. Both attenuated the anti-nociceptive effect of Ozanimod. In practical terms, that suggests Ozanimod's beneficial effect in the RVM engages serotonergic and noradrenergic inhibitory circuits that act at the spinal level. This is clinically meaningful because it frames neuropathic pain not only as excess incoming nociceptive drive, but also as impaired endogenous inhibition. In other words, the paper supports the idea that part of the disease may be failure of the descending brake. If a treatment can help restore that brake, that could complement our current ways of thinking about neuropathic pain management. Another subtle but important result is what did not happen. The authors injected a selective S1PR1 agonist, SEW2871, into the RVM of uninjured animals, and this did not induce pain-like hypersensitivity. That suggests the role of S1PR1 in the RVM is state-dependent. In a healthy system, activation of receptor may not be enough to create pain behavior on its own. But in an injury state, the pathway may become pathologically relevant. That kind of context dependence is often what we see in chronic pain biology, and it may be important for therapeutic selectivity. There are also several limitations that should keep our interpretation balanced. First, this is an animal study, and all experiments were done in male rodents. The authors know prior work suggesting similar responses across sexes in related models, but sex was not directly tested here. Second, the behavioral outcomes are measures of evoked hypersensitivity, mainly mechanical and cold allodynia. Those are standard and useful, but they do not fully capture the multidimensional patient experience of neuropathic pain. Third, while the RVM data are compelling, the exact cellular targets remain uncertain. S1PR1 is broadly expressed in the nervous system, including glial population, and the study does not fully resolve which cell types are most critical in the RVM. Fourth, because Ozanimod was also effective systemically, the overall analgesic effect in a living organism may reflect combined peripheral, spinal, and supraspinal actions. So what is the take-home message from this paper? I would say it is this. Ozanimod, an FDA-approved S1PR1 modulator, reduced neuropathic pain-like behavior in multiple rodent models, and this study provides evidence that one important site of action may be the rostral ventral medulla, where S1PR1 signaling appears to disrupt descending inhibitory pain control. The associated normalization of type 1 interferon-related gene expression as an interesting mechanistic layer, although that pathway still needs further causal clarification. For clinicians and pain researchers, the broader implication is that brainstem descending modulation remains a very important therapeutic frontier. Neuropathic pain is not only a peripheral nerve problem and not only a spinal cord problem. It is also a network problem. This paper suggests that targeting S1PR1 may help rebalance that network. That is an exciting idea, but it should still be interpreted cautiously until we have human data, specifically addressing neuropathic pain. Even so, as a translational paper, I think this study is thoughtful, mechanistically rich, and clinically provocative in the best sense of the word. Thank you for watching.